So this is just a continuation of this lecture. I chunked it into elements that are easier to maintain attention, because shorter bits. But the learning objectives are the same as the last lecture. This is just a second chunked part B, if you will. But there are several other interconnected gray matter structures buried into brain regions that contribute to an array of behaviors. And again, it's very important, I think, to think of these things as interconnected loops rather than discrete brain regions that operate in and of themselves. So the basal ganglia, this is something that we've already alluded to a little bit, but the basal ganglia is very important for motor control. The cognitive cortical subcortical loops that I demonstrated before in last lecture are basically the basal ganglia. It's the basal ganglia interacting with the cortex in these cortical subcortical loops, or basal ganglia cortical loop, if you will. This is very important in motor control, development of habitual motor sequences. Clive Wearing, I think, is a great example here. Clive Wearing was a famous composer. He put on the show for Princess Diana's wedding. So he was like a big, prominent, well-known guy. And he also, had quite the social life, ended up with syphilis, unfortunately, which destroyed part of his brain, but it only destroyed parts of his brain like the hippocampus and regions of the brain that are very important for associative memories and spatial memory. His basal ganglia remained intact. And what's amazing is that he could compose new piano pieces and remember them, be able to play them. So he could just sit down at the piano and play some piece that he just composed, but he wouldn't remember writing it because his basal ganglia was not only, intact, it was a fantastic basal ganglia. And I also like to think about these things in terms of intelligence a little bit. What is intelligence? What about basal ganglia based intelligence? You might say, yeah, so what? But we actually highly value this in America, don't we? This would also be involved in what we think of as muscle memory. So, we not only in the form of a piano player, but what do we value quite a bit in America? It's sports, athletics. So like Barry Sanders, the famous running back, he probably had a basal ganglia that borderlined on savantism. He could see the field and break things down and anticipate where he needed to go, a split second faster than anybody else on the field. A lot of it was probably, basal ganglia associated. One region of the basal ganglia is the caudate putamen, what I also refer to as the dorsal striatum. This is involved in coupling emotional information with motoric output. So, you can see how that would be very involved in the basal ganglia as well. The substantia nigra contains dopamine neurons that innervate the caudate and putamen and sort of send the dopamine signals to this region. This would be very important for movement. It is, and interestingly, these neurons have a myelin-based black pigment that is why they're named the substantia nigra. And that would be the compacta, not the reticulata, technically a distinct brain region that is also a part of the basal ganglia. The thalamus is technically not in the diencephalon, not the telencephalon, but it's very important as it's interconnected here and relays sensory and motor inputs to the cortex. It's sort of thought of as like the main switchboard, the main relay center of the brain. So most information that, for example, would be occurring in our body that might affect everything, like our emotions, we've heard countless things. It's not just the GI, like sending signals to the brain. It's all throughout our periphery. It's not just the gut. That's important, but all like the immune responses, everything throughout the body comes up to alter emotionality. If we're in chronic pain, like your leg is in chronic pain, that is going to influence how we feel pain and everything. All experience will be changed. But a lot of that relay to the cortex occurs through the thalamus. So this is sort of like a relay hub. And all of these regions are connected or interconnected through loops or tracks. So a few other regions that I want to just briefly go over. Again, this is a review from 2220. The limbic system is a general way of describing a variety of brain regions involved in emotions and learning. Key structures of the limbic system include the amygdala, so named because it looks like an almond, that is very known to regulate emotion. In particular, we will study how it is involved in fear memory. Of course, we'll also talk a little bit about how it sort of fits into a larger circuit to modulate fear memory. But the amygdala certainly plays a central role in fear memory expression. The hippocampus, so named because it looks like a seahorse, regulates learning and memory, certain kinds of learning and memory, not so much the procedural memory that we talked about with Clive Waring, but more associative and spatial memory. The cingulate gyrus is very involved in attention and higher order cognitive executive function. And the olfactory bulb is involved in the sense of smell. And it's also worth pointing out that smells, in part because of its interconnection with other parts of the limbic system, can be associated with specific memories, whether it be joyous memories or traumatic ones. And the smell alone can bring back a lot of these memories and sometimes function as a trigger, hopefully a positive one more than a negative, but certainly both. A lot of what we know from the brain has come from observations of what happens when a region of the brain is damaged. One good example here for the amygdala is something known as Cliver-Bucy syndrome. This results from bilateral destruction of the amygdala. So what happens if you get rid of this? This was first seen in animal studies following experimental ablation. You just go in and lesion, typically either electrically or chemically lesion a brain region to see what happens. But it also occurs in humans. It's occurred through following certain lobotomies. And it can also occur following severe viral infections of the brain or following stroke, which, as you probably know, is when blood is where it should not be in the brain, basically a brain bleed. But what happens if you destroy the amygdala is that you see an increase in sexual activity. Subjects tend to compulsively put things in their mouths. Sort of an oral fixation. You see overall decreases in emotionality and changes in eating behavior and visual agnosias. For the hippocampus, a lot of our initial understanding of this region came from a study of a very famous patient in psychology known as H.M. In 1950, Henry Molaison underwent radical surgery in an attempt to stop his epileptic seizures. His doctor, William Scoville, removed both sides of his hippocampus despite having little knowledge about what the hippocampus does. When Henry recovered, he could remember some things before the surgery, but it seemed he could not make new memories. A psychologist named Brenda Milner was brought in to study Henry. She found that Henry couldn't form new declarative memories. These means facts, things we saw, people we met, common associations. He would forget them instantly. But his ability to form procedural memories, similar to Clive Waring, was still intact. He could learn new skills, he just couldn't remember learning them. He would forget them instantly. Because of these studies, we now know that these two types of memories are stored in different regions of the brain, and that the hippocampus acts like a switchboard, linking together the different storage networks in the brain. I mean, really, if we play that analogy out too much, everything in the brain is a switchboard. But, anyway, unforgettable cases like Henry's helped advance our understanding of the brain. He died in 2008 at the age of 82, though in his mind, he was still 27. So now let's move on to the diencephalon, which is a division of the forebrain situated between the telencephalon, which you've already discussed a little bit, and the midbrain. And we've already sort of dabbled in a few brain regions and circuitry that interact with the diencephalon. And again, it's really, I think, difficult to think about these regions without being interconnected to some degree. So the diencephalon, two of the main structures there are the thalamus and the hypothalamus. We've already described the thalamus as this sort of central relay, the major switchbox for motor and sensory information. I've already pointed out that, a lot of brain regions can be thought of as switch boxes. This is particularly true with the thalamus. Not only does it do that, it seems like that's sort of its primary job, just to sort of function as a sort of big relay hub. The hypothalamus, located under the thalamus, which is the basis of its meaning, is very different in function. It specifically is related to endocrine function or hormonal function. And we are going to go over that in some detail later in the class. But it controls many of the sympathetic nervous system responses through the pituitary gland, which would then secrete the actual hormones. It is most commonly associated with the 4 F's, which are fighting, fleeing, feeding, and fornicating. So this brings us to the mesencephalon, which is also called the midbrain, which is how I typically refer to it, just a little simpler on the tongue. But the midbrain contains the dopamine neurons that project to the dorsal striatum, or the caudate and the putamen. The mesencephalon is also known to connect the brainstem to the forebrain. So, it is very important, and especially as a dopamine researcher, I see it as being critically important. 2 regions of the mesencephalon or midbrain that I want you to be familiar with are the substantia nigra and the reticular formation. I've already mentioned the substantia nigra pars compacta. That is the origin of most of the dopamine neurons that then go on to the caudate putamen. It innervates the dopamine or the basal ganglia with dopamine, and importantly, these cells die in Parkinson's disease, resulting in motor dysfunction. This is something we'll talk about to a great degree later, but again, think about this real quickly, just briefly, in terms of the circuit. It's not just the loss of the dopamine neurons that causes tremor, right? So, really, what has to happen there to see the tremor development is a whole cascade of circuitry items. So you have to lose about 80 to 90% of the dopamine neurons in your midbrain. They have tremendous compensatory power. The brain doesn't jump. There are a lot of redundancies built in to help us deal with neural injury. But you have to lose about 80 to 90% of your dopamine neurons before you really even see Parkinson's emerge. And then it's not like it just happens out of the blue because there's a little less dopamine greasing the wheel in the midbrain. It doesn't work that way. So what happens is you have a loss of dopamine input into the primarily GABAergic neurons that make up the dorsal striatum. Then these output along 2 separate paths known as the direct and indirect pathway. At least this is still, for the most part, accepted for the dorsal striatum. These innervate separate portions of the globus pallidus and then come back together eventually in the thalamus, that relay again. Interestingly, they come in sort of differently at that point. They're converging differently. And there's a whole bunch of complexity that goes on there. But then at that point, this message would continue on to the basal ganglia. To the motor cortex, the primary cortex, which would then send the signal down to your limbs to move. And the entire sort of the series, the circuitry is what is impaired. The ability of, the globus pallidal structures to properly integrate back with the thalamus and then send on the proper signal to the motor cortex is all disrupted when you take out this one domino. But that's what's going on here. It's like a domino effect. It's not just, dopamine not, greasing the wheel in the midbrain. The reticular formation is also fascinating and worth bringing up. This extends from the midbrain to the hindbrain and is involved in many behaviors, including sleep, arousal, temperature regulation, and motor control. The mesencephalon comes next. This is the embryonic part of the hindbrain that differentiates into the pons and cerebellum. So again, we're starting in this one tube, and this is the part that folds in to make the pons and the cerebellum, but it also is the seat of several cranial nerves, including the trigeminal, abducens, facial, and part of the vestibulocochlear. The pons is the origin of several cranial nerves, like I already alluded to. And also the rostral portion of the brainstem. So, it's involved in very many of our basic functions. I've already mentioned the respiratory center in the medulla, which is just ventral or, I guess caudal, depending on what sort of species we're talking about, to the pons. The medulla actually sends those respiratory signals to the diaphragm. But if there were going to be an increase or decrease in action potential output to the diaphragm, that comes from supplementary respiratory centers in the pons. The cerebellum is just absolutely a crazy structure. I mean, like to put it bluntly, we typically think of it as being involved in fine motor control, but it is much more than that. Alcoholic gait, the inability to pass the sobriety test is due to temporarily, hopefully temporarily, dysfunction to the cerebellum. However, we now recognize that the cerebellum is involved in learning and memory, and some even go as far to call it a little brain and propose the existence of cerebellar loops with the rest of the brain that may be important for very complex tasks. I mean, so the idea of the cerebellum just being this fine motor structure is, selling it very short, I'd say. It's probably involved in very, complex function, and some have even suggested that evolutionarily, maybe it played a, role similar to what our cerebral cortex has evolved into, earlier in evolution, or maybe in certain species. And finally, our last embryonic developmental region that all originated with that tube, the tail end of it, the myelencephalon, primarily made up of the medulla. Incredibly important, structure of the brain, as are really all of them. But, we would die without the medulla because, our diaphragm would stop being innervated, all of our trunk functions would cease to occur. So the medulla, of course, is involved in so many critical, vital functions for, just our overall, unconscious bodily function. This caudal region or ventral region of the brainstem, again, depending on if you're talking about a rat or a human, marks the transition to the spinal cord and is the source of many cranial nerves, including those that go to the tongue, amongst other things. I've already mentioned the respiratory groups of the medulla send signals to the diaphragm through the parasympathetic autonomic nervous system to maintain respiration. You might also be interested to know, what happens if the medulla is damaged. First of all, you can die. There are opioid receptors in this region that are known to suppress neural activity if there's too much opioid on board. Interestingly, cannabinoids do not have receptors in this brain region, which is one distinction between opioids and cannabinoids. A very important one that can mean life or death. But, the medulla also, if it's damaged, a variety of other things can occur in addition to death. Some more bizarre things can happen. For example, if medullary damage can result in the loss of the sense of touch. So, you can't sense the ability to feel anything. It's such an important area in the brain. You're going to learn about how various input to the cortex through the thalamus are relayed through the medulla. Some of this information synapses there. And if that is lost, you lose the sense of sensation. Another one that can happen, or that has been reported, is that the patient can lose their sense of position or not know where certain body parts are relative to others. So, I think these just point out how critical the medulla is, and it's such a base structure too. I also think of it as when we think of complexity of human behavior. And again, it's not it's so different than that of a non-human primate or a cat even. What there is a more advanced sort of functionality that emerges, right? Now, but the medulla, I think of it as sort of the core base that a lot of the mammalian brains are built on. And then the level of complexity sort of builds out as you go. But the medulla just very critically, very evolutionarily conserved with a lot of these just like vital functions like breathing.